My name is Caleb Dunn, and today we're going to talk about antibody-mediated effector functions. So there's four ways that the antibodies in your body can protect yourself against pathogens. And this is through neutralization, opsonization, and the third one, complement fixation, and the last one is called antibody-dependent cell-mediated cytotoxicity, but we'll call it ADCC for short. So we'll start by talking about how antibodies can neutralize pathogens, and this is when you have antibodies, and then you have an antigen. And your antibodies are going to bind at a specific site on the antigen. And what this is going to do is this is actually going to mask the antigen. So in short, this is going to keep the pathogen from being able to enter the host. So once this happens, there will be a phagocyte that will come in and it will engulf these complexes that have been formed. And these antibodies, they're called neutralizing antibodies, and these can be used to prevent toxins from entering the body. And an example of this would be, say, tetanus trying to enter the body. This disease can cause uncontrolled muscle response and death, but the tetanus vaccine can be introduced to the body, and B cells can be produced to fight against this pathogen. And the antibodies produced in this case would bind to and stop the entry of the toxin into the cells. This type of effector function is also used to fight snake bites. So the second type of antibody-mediated effector function is called opsonization. And this is when the antibody will bind to a pathogen. So again, you have antibodies, and they will bind to a specific pathogen. So now they have bound to a specific pathogen. And now this complex is going to meet a phagocytic cell, and they're going to link up by what's called the Fc receptor of phagocytic cells. So now this Fc region of the antibody is going to bind to these Fc receptors on the phagocytic cells. And once this happens, the phagocyte will destroy the antibody-antigen complex that's been made once it binds to this Fc region. So it's going to induce phagocytosis on the complex. So there's ways that opsonization can be defective. People have diseases that can cause deficiencies in the complement system. This will make them more prone to infections, and more specifically bacterial infections. So if you have a defect in the complement system, you may be more likely to have an infection that wouldn't normally affect a healthy person. And the third way that antibodies can fight pathogens is by complement fixation. Now, in the case of this, this is going to be when an antibody binds to an antigen, and it's going to create a whole cascade of events called the complement cascade. So the complement cascade is when an antibody and an antigen are going to form a complex called the C1 complex, and this complex will form different fragments called C2A and C4B fragments. Now these fragments will combine together to form what's called C3 convertase, and this will cause C3 hydrolysis. So once this happens, it's going to form C3b and C3a fragments, while the C3b will cleave C5 into C5a and C5b fragments, and the B fragment of C5, it'll combine with C6 through C9 complexes, and it will form MAC, or the membrane attack complex. And this is going to destroy the pathogen by phagocytosis or lysis of the cell. And there's one more function, which is called antibody-dependent cell-mediated cytotoxicity, or ADCC. And this particular function is pretty similar to opsonization in that you'll have antibodies and they will bind to a specific antigen. Just like this, they'll bind to this antigen, and they will bind to an Fc receptor on a cell. But this one is a little bit different. Rather than using a phagocyte, this actually going to use more specifically cytotoxic T cells, or even more specific, it's going to use natural killer cells in this case. So, like I said, this will begin when the antibodies will bind to antigens, and then these natural killer cells that are floating around, they can recognize this complex that is formed by the Fc receptors on this antigen-antibody complex. So these two will cross-link, and this will trigger the signal for apoptosis. Now these complexes have committed apoptosis, and the pathogens will die. So this type of function can be used as a form of cancer treatment, as monoclonal antibodies can be used to take advantage of ADCC, and it can direct natural killer cytotoxicity toward tumor cells that might be in the body. And those are the 4 antibody-mediated cell effector functions.